We're going to continue to talk about other nucleophilic acyl substitution reactions, other reactions of carboxylic acid derivatives, focusing on biological systems. Now, if we think about nucleophilic acyl substitution of carboxylic acids themselves, they're not very good. So they need to be activated. And so in biological systems, a carboxylic acid can be activated by reaction with ATP. And so this is going to add to this phosphorus, and it does basically nucleophilic acyl substitution, where this adds here. These electrons go up, back down, kick this out as a leaving group, giving us the acid with a really good leaving group. Now, a nucleophile such as coenzyme A can react here, push these electrons up in a nucleophilic acyl substitution, comes back down, kicks out this whole entire leaving group to give acetyl-CoA. And acetyl-CoA, this is an example of a thioester. This is involved in about 1 million different biochemical pathways. Now let's look at the same reaction, but from the perspective of acetyl-CoA. So this whole thing is coenzyme A, and then we have this thiol at the end of it, and then that can react with the acetyl adenosyl phosphate that we saw on the last slide. So this sulfur is the one that will add here, push the electrons up, back down, kick it out, and that puts this acetyl group on this molecule. Here we have some of the reactions that are involved in the biosynthesis of many different types of compounds. So here we have acetyl-CoA, and this can do a nucleophilic acyl substitution. It's really a trans-esterification, where this is going to add here, up, down, kick this out, so that we make this acetyl-ACP, where ACP is the acyl carrier protein. A different reaction that can happen is that acetyl-CoA can be converted to malonyl-CoA. And here there is an enzyme that can act as a base, and it grabs off this proton, and then this is going to add to carbon dioxide, giving malonyl-CoA, where this group has been added on at this position. So this is malonyl-CoA. This can be hydrolyzed to give malonate, or this can react with the ACP thiol to give malonyl ACP, which is the same as acetyl ACP except a little bit bigger. This can react again to give acetoacetyl ACP. This is basically a Claisen condensation that we're going to learn about in the next chapter, giving us a different thioester. And then this can be reduced, and then it undergoes elimination, and then another reduction to give this, which is called butyryl acyl carrier protein. So who cares about this? Let's look at the next slide. This compound, the acetoacetyl ACP, this is converted in a few steps to IPP, which is called isopentenyl pyrophosphate, and IPP is in equilibrium with DMAPP, dimethyl allyl pyrophosphate. And both of these are involved in the biosynthesis of terpenes. Terpenes are a big family of compounds that all have the isoprene unit, which basically is this 5-carbon piece. And so there's a lot of methyl branches around, for example, and there are a lot of natural products that are terpenes. And here we have a few examples. This is menthol, which is in peppermint. This is carvone, which we worked with in lab, and so one enantiomer is spearmint and the other one is caraway. This is camphor, so this is another terpenoid, and this is Vicks VapoRub. Right? A lot of spices have terpenes, so caryophyllene, the Pac-Man compound, this is a major component in the essential oil of cloves. In addition, we have other compounds like this one, geraniol. This is in roses. We have vitamin A is also biosynthesized through IPP and DMAPP. Beta carotene, that's important for vision. Squalene is another important compound, and squalene can be converted into cholesterol, which is then converted into various hormones as well as steroids that are responsible for many important regulation processes in the body. And all of this is based on organic chemistry. Also related to that sequence of reactions is the biosynthesis of fatty acids. Fatty acids always come in even numbers because they are biosynthesized in 2 carbon pieces. And so the butyryl ACP will come back and react with the malonyl ACP. It'll continue going through this, forming a variety of fatty acids. Now, the fatty acids can then react with glycerol to form triacyl glycerols. So these are fats and oils. These are responsible for energy storage. They can be converted into phospholipids, and these are important components of cell membranes. They can be converted into waxes, like this compound. These are important on leaves or fur and feathers. They offer water-repellent protective coatings. In addition, specifically from a C20 fatty acid called eicosanoic acid, there's a variety of eicosanoids. So this is a prostaglandin, and prostaglandins are important chemical messengers. This is thromboxane. This is responsible for platelet aggregation in blood clotting. And this one is called leukotriene. This is responsible for the constriction of bronchial passages during asthma attacks. It's a lot of the reactions that we have talked about, that we're going to talk about, that are responsible for the biochemical pathways, the biosynthetic pathways that lead to incredibly important molecules. So nucleophilic acyl substitution is key in the biosynthesis of various lipids that are important for structure, for energy storage, for cell membranes, for coatings, as well as hormones, including the sex hormones, as well as the biosynthesis of chemical messengers that are responsible for controlling things like fertility and conception, heart rate, metabolism of carbohydrates, lipids, fats, as well as allergic responses. So this concludes Chapter 21 on carboxylic acid derivatives and nucleophilic acyl substitution reactions. I want you to be familiar with general information about carboxylic acid derivatives, be able to identify and name carboxylic acid derivatives, be able to compare their physical properties, be able to identify them using spectroscopic methods, and know about ways to prepare them other than through nucleophilic acyl substitution, as well as through nucleophilic acyl substitution. I want you to know the general information and then think about the interconversions, what works, what doesn't work. Know the big picture of the mechanism in terms of formation of the tetrahedral intermediate and dissociation of the tetrahedral intermediate, and also be able to work through the details to provide a stepwise mechanism for particular reactions, such as ester hydrolysis. And finally, I want you to be able to consider more broadly other types of reactions of carboxylic acids and carboxylic acid derivatives. So how are they prepared and how do they react? And then finally, just to recognize their important role in biological systems as well.